Welcome technician, continuing with our NABH related activities. So next important thing is the infection control practice. So one of the most important thing regarding the infection control practice is the hand hygiene. Hand hygiene, biomedical waste management, these are all part of the universal precautions. So in that, hand hygiene is one of the most important infection control practice. Okay, there are two types: hand wash and hand rub. Hand wash where you use a soap solution, hand rub where you use a alcohol based solution. Okay, so there are 5 moments in hand hygiene. This is mainly with respect to patients. For the lab it doesn't apply so much, but still it's always better to know. So what are the 5 moments of hand hygiene? Before touching the patient, before any procedure like BP taking or some IV cannula applying or anything of that sort. So any before procedure, after that particular procedure, or after you are exposed to some body fluids or something, and then after touching the patient and after touching the patient surroundings. These are all the 5 important moments of hand hygiene. Okay, how to do the hand rub? So for doing the hand rub, first we will first check with the hand rub. Okay, if for hand rub, just make a little cupping of your hand and put all the hand rub solution in your hand. Okay, so once you hand, then first to palm, okay, rub palm to palm, and then interlacing of the fingers, that is first right hand above, then left hand above, right hand above, and then left hand above, and then finger interlaced, so in the like that, and then followed by back to back fingers with opposing palms, then knuckles, okay, thumb and the thumb part, and then again with the fingers with the palm, then ultimately allow it to dry first. So whole procedure should last not more than 30 seconds. Okay, it should be over within 30 seconds. So this is the for every hand wash sink we have put the hand rub and hand wash steps. So even in your sleep you should able to do this. Okay, now how to do hand wash? So the first important thing about the hand wash is, first rinse your hand with water and then apply the hand wash solution. Once you do put the hand wash solution, again the same, first palm to palm, then the right hand upper dorsum of the hand with the finger interlaced, then left interface, and then within the opposite palm interlaced, then opposing palms with finger interlocked, and then through the thumb, and then with the fingers, and again wash the soap with water. Allow it, use the disposable wash towels. Okay, you can use a tissue paper and then allow the hand to get dried. So the diagrams are there which are more than self-explanatory, so you can understand it. Again you have to know. Now what is the difference between a hand rub and a hand wash? So hand rub is done when the hands are not stained or dirty or exposed to any body fluids or anything of that sort. Okay, it is done only in between. Okay, it's like a emergency procedure like that. At the most continuously you can do hand rub 5 times. Okay, from morning till evening you can't keep on doing only hand rub, so you can do hand rub 5 times. After that it should be followed by hand wash only. If your hand is visibly dirty or any body fluids or anything has touched, then you should go for a hand wash only, not hand rub. Okay, no hand rubbing or hand washing on a gloves. So some people have that habit, they wear a gloves and on the gloves only they do hand rub and hand wash. That should never be done. Okay, these are all some important things that you should know regarding hand rub and hand wash. Next important thing is the biomedical waste management. So what to put in where? So segregation of the biomedical waste at the site of production itself is one very important thing. So first and the foremost thing is the black colored bin. Black colored bin is used for all type of general waste, plastics around the reagents, the plastic container that comes with the reagent, or the cardboards which comes with the reagent. So all these things will go for general waste. Okay, green colored bins are mainly used for food items, organic waste and other things and all. So black and green colored bins and all, they will go to the general to the part of the municipality. Okay, they are the ones who are responsible. Whereas the red, yellow and white color, red, yellow, blue and white colored and all, they all go to the biomedical waste department. Okay, so red colored bins, this is the general guidelines. So wherever there is any where the plastic material is there which is infective and all, it will go to red colored bins, because when you burn the plastic, it releases some gases which is not good for the environment and all. So it all goes to red colored bins. Whereas the yellow colored bins, this is also infective waste, but it can be burnt, like cotton and all. So it can be burnt, anatomical part and all, it can be burnt. So they all goes to yellow colored bins. Okay, blue colored bins, in further lab, all these glasses which are broken, like glass slides, ampules, or maybe a beaker or something, glass that has broken. So all these things goes to blue colored, and this blue colored bin needs to be puncture proof. They are not plastic covers. Okay, they are puncture proof containers. Okay, white colored bins, all sharps and needles will go to white colored bin. So if a syringe is given, the needle should go to white colored puncture proof, whereas the syringe body per se will go to the red colored ones. So like that. So this is the whole thing by which it's a general understanding. So if you go to the red colored bins, so you have tubes. So cotton per se is not put in red colored bins. Whereas the IV tubes, IV bottles, IV sets, catheters, urine bags, syringe without needle, vacutainer gloves, all these things will go to red colored bins. Whereas in yellow colored bins, it's a cotton will go here, not in the red colored ones. Cotton will go to the yellow colored bandages, swabs, dressing, caps, masks, aprons, diapers, sanitary napkins, then the blood and body fluids, chemical liquid waste. So all microbiological related waste, all these things go to yellow colored bins. Whereas in blue colored bins, the glasswares, ampules and vials, any implants of the patient, slides, broken glass, all these things go to blue colored bins. Whereas in case of white colored bins, it is the needles, blades, scalpels, sharp metals, lancets, these items will go to white colored bins. Okay, black color is for general waste. So in the lab, what are the items where it goes? This is very important, we should know. First and the foremost in vacutainers, the used vacutainers which contains blood or any other things and all, they will all be with the cap, they are sent to CSST for terminal sterilization. Okay, after 48 hours of keeping them for any retest or anything is required, after 48 hours we send all these vacutainers into a red colored bins. Urine containers, when they contain urine inside them, they all will be emptied into the commode, and the container is put in the red colored bins. Okay, stool containers, the whole stool containers should be sent to CSST lab after adding hypochlorite solutions of equal amount. Okay, all this rapid CAR test, HIV, HBsAg, HCV CAR test and all, you put a 2 drop of hypochlorite solution on the sample area only, and then put them into red colored bins. Microbiological swabs, the swabs which you use for doing antibiotic sensitivity and other things and all, they will go to yellow colored bins. Whereas the patient swabs which comes with wound swabs and those type of swabs which has got a plastic cover around it, they all goes to red colored bins. All the cotton waste that is used in our lab, they all go to yellow colored bins. Reagents and chemicals, whether it are plastic ones or not ones, all the smaller reagents and chemicals like our C311 instrument ka, and then our E411 ka instrument ka, so all those things will go to yellow colored bins only. Okay, whereas the microbiological culture plates and all, they all directly will send to CSST for terminal sterilization. Okay, so then blood bags, unused blood bags, whether it is packed cells, FFP, or platelets or cryoprecipitate, all these things are directly sent to CSST for terminal sterilization. Whereas large plastic containers like ABX diluent, ABX cleaner solution, then our reagents from the HbA1c instrument and all, so all these things are, after emptying, they are sent to the store for either management from themselves. Okay, all the liquid waste that gets generated, like from hematology instrument, the liquid waste gets generated, from biochemistry instrument, liquid waste gets generated. All these things are put into a sink in the next to our distilled water plant. So it is emptied directly into it, because from there the pipe will go directly to the treatment area in the hospital. So it is dumped directly into that wash basin. Okay, so this is how all the things are biomedical waste that are generated in the lab is taken care of. Hope you will be able to understand it properly. Thank you.